Hey everyone, Dr. D here. Let's continue on with Chapter 9 from our Biology Concepts and Investigations textbook from Hoefnagels. Let's go ahead and get started.
Dr. D, 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 Dr. D. Explain stuff.
All right, welcome back. Let's go ahead and get started with Chapter 9, which covers sexual reproduction and meiosis. In the previous chapter, Chapter 8, we learned that reproduction can be asexual. So this means that during asexual reproduction, one parent organism replicates its DNA and splits the contents of one cell into two. This results in two genetically identical offspring. So for instance, if this is an original cell and this cell undergoes mitosis to form two cells, those two cells are identical cells. This is a form of asexual reproduction. This is actually an amoeba. This amoeba is reproducing asexually by mitosis. If a single cell eukaryote replicates by mitosis, that's known as asexual reproduction. And if a prokaryote, do you guys remember prokaryotes include bacteria and archaea? If a prokaryote replicates, that's known as binary fission. Remember that? And that's also a form of asexual reproduction. In all, you should know that asexual reproduction results in two genetically identical cells. But that's not what we're here to learn in this chapter. In this chapter, we're going to learn about sexual reproduction, and this involves meiosis in order to produce the sperm and the egg, which are known as the gamete cells. And then when the sperm and the egg, come together during fertilization, that's a form of sexual reproduction, and that results in genetically different offspring. Okay, so that's what we're here to learn about now. So again, in sexual reproduction, the DNA of the offspring comes from two parents, an egg from one parent and a sperm from another parent, yielding the first cell of the next generation. This is known as a zygote. This results in genetically diverse offspring. Genetically diverse offspring. So the question at hand is, why do sexual reproduction? Attracting a mate and producing sex cells requires a lot of energy, which is costly to the organism. Why is sexual reproduction so common, and why is it so important? Why can't we just clone ourselves? Why can't we just undergo asexual reproduction all the time? That saves a lot of energy and a lot of resources. So what's the benefit of sexual reproduction? Let's touch on that next. Well, it all comes down to survival. Genetic diversity is worth it. Sexual reproduction produces genetic diversity among your offspring, which increases the chance that some offspring survive in a changing environment. So, for instance, if there's only a little genetic diversity, over time, there might be an ecological disaster, such as a new virus that comes along or something happens in the environment. And this can cause every organism to perish, right? However, when there's genetic diversity, then you might have some variants of your population that are able to, carry on because the ecological disaster might um, might take away these members of the population. However, due to that genetic diversity, others may survive and carry on the lineage, right? So genetic diverse populations are more resistant to ecological disasters and more likely to persist over time. This makes sexual reproduction well worth it because it makes it so that it's far less likely that a species goes extinct. Now, this leads us to clicker question number 1. Which two organisms are genetically the most similar? Pair A, which was the amoeba after it underwent asexual reproduction by mitosis, or these two pups, which were the product of sexual reproduction. Let's think about that and see if you can beat Wicket. That's right, Wicket. Pair A. Pair A are genetically more similar. In fact, they should be genetically identical, right? Because those two amoeba formed by asexual reproduction. So it should be pair A. Okay, whereas pair B came from sexual reproduction. And so these two pups should be genetically different. Now let's talk about how the chromosomes work inside of sexually reproducing organisms like this dog here and its pups. In a sexually reproducing organisms have two sets of chromosomes. And these cells are called diploid cells because they have two sets of chromosomes. So now let me more clearly explain what do we mean by diploid cells and what is a set of chromosomes and why do, animals like you and me, why do organisms that do sexual reproduction, why do they have two sets of chromosomes? So let's talk about that next. So take a look at this. On the right is what's known as a karyotype. A karyotype shows all of the chromosomes from a diploid human cell. How many chromosomes do humans have typically? Typically, how many chromosomes does a human cell possess? Can you beat Wicket? That's right, Wicket. 46 chromosomes, right? A typical human cell contains 46 chromosomes. That's because our entire genome is made up of 46 chromosomes. But did you know that there are only 23 different chromosomes? There's chromosome 1, chromosome 2, chromosome 3, chromosome 4, etc., all the way up to chromosome 23, which are known as the sex chromosomes, X and Y or X and X. Okay, there's only 23 different chromosomes. And did you know you inherited one set of 23 from the egg, from your mom, and one set of chromosomes from the sperm from your dad, right? And so that's what they mean by a set. There are 23 different chromosomes, 1 through 23, and you inherited one set of 23, so a chromosome 1, 2, 3, chromosome 1 through 23 from the mom, from the egg, and a chromosome 1 through 23 from the sperm, from the dad, right? And so if you look at it here, that's why you, in this karyotype, you can see that a human cell would have two chromosome 1s. One's from mom and one's from dad. And you also would have two chromosome 2s. One's from mom, one's from dad. Two chromosome 3s, one's from mom, one's from dad. And these are known as homologous chromosomes. So it's very, very important that you keep track of these terms, homologous chromosomes versus sister chromatids. Homologous chromosomes are your two different chromosome 1s, right? One from mom, that's known as the maternal chromosome, and one from dad, known as the paternal chromosome. Now, let me see if you can beat Wicket. Are these identical chromosomes? Are they identical genetically? No, that's right, Wicket. They shouldn't be identical because one's from your mom and one's from your dad, right? They're both called chromosome 1, but this is chromosome 1 from mom, the maternal chromosome 1, and this is chromosome 1 from dad, just like this is chromosome 2 from your mom and chromosome 2 from your dad, and so on and so forth. So homologous chromosomes, what do they share? Homologous chromosomes share the same chromosome number, but they are not genetically identical. I want you to write that down. Homologous chromosomes share the same chromosome number, but they are not identical. You inherited one from the sperm and one from the egg. So you might be wondering, well, what do they have in common? Why are they both called chromosome 1? Well, this is what you need to jot down. They have the same types of genes, right? So for instance, I'm just making stuff up. If the hair color gene is on your mom's chromosome 1 it would also be on your dad's chromosome 1. Does that make sense? So hair color would be here, but does that mean that you inherited the exact same hair color information from both parents? Oh, right, Wicket. You would not inherit the same unless they both had brown hair, right? You could have brown and brown, right? You inherit the brown gene from your mom and the brown hair color gene from your dad. But couldn't you inherit, like, let's say, the blonde hair color gene from the mom and brown hair color gene from dad? You see, so the same types of genes are found on the chromosomes, like eye color, hair color, etc. But they're not the exact same DNA, are they? Because they're from different parents. So again, you should know that in a human, we have 46 chromosomes. That's because we have two sets of 23, one set from the mom, one set from the dad. These are known as homologous chromosomes, homologous sets of chromosomes. They share the same chromosome number and the same types of genes on those chromosomes, but they are not identical because they are from different parents. So hopefully that makes sense because chapter 8 and 9 really are made easy once you understand the difference between homologous chromosomes, which are not identical, and sister chromatid pairs, which are identical. Okay, so keep those in mind, and these chapters are going to be so much easier to understand. By the way, there are, again, 23 different chromosomes. And remember, I told you that the 23rd pair are the sex chromosomes XX or XY. Okay. The rest of the chromosomes, chromosomes 1 through 22, are called autosomes. Autosomes. These are the same in both males and females. Whereas with the sex chromosomes, you typically have two X chromosomes for females and an X and a Y chromosome for males. Now let me give you an example. Let's pretend we're focused in on chromosome 1 and we're looking at the two chromosome 1s inside of your cells. Remember, these are called homologous chromosomes. One's from mom, one's from dad. And they share the same chromosome number and the same types of genes. So let's imagine this pair right here. Let's look at these in more detail. So take a look. Here it is. Imagine if this was chromosome 1 from your mom on the left and chromosome 1 from your dad on the right. These are both chromosome 1s. So if they are both chromosome 1s, but one's from each parent, that makes these homologous pairs, homologous chromosomes. Now, remember when I said the same types of genes? So that would be gene A and B and D. You see these genes A, B, and D? I'll just make up something. Let's say gene A dictates your hair color. Gene B dictates your eye color, right? So hair color is A, eye color is B. Now again, does that both chromosome 1s, right? Both chromosome 1s would have those genes for hair color A and eye color B. But does that mean they have to be the exact same hair color, the exact same DNA sequence? Can you beat Wicket? No, that's right, Wicket. No, they can be different, right? So for instance, if capital A means brown hair color, that means you inherited brown hair color from mom, brown hair color from dad. However, for eye color, maybe a big B is brown eyes, but the little b represents blue eyes. So you inherited blue eye color gene from dad and a brown eye color gene from mom. You see how it's the same type of gene, eye color, but you could inherit brown eye color from one parent and blue eye color from a different parent? That's what I'm talking about with regard to these homologous chromosomes. Now, real quick, you know, uh, again, the gene would be called what? The gene would be called hair color or eye color. But what is the term for different versions of a gene? So, for instance, brown eye color gene versus blue eye color gene? Well, the term for that, you need to write this down. The term for that is alleles. So when I say an allele, I'm talking about a version of a gene. So if the gene is eye color, then alleles would be blue and, oh, sorry, brown and blue, right? Does that make sense? Again, if the gene is called eye color, the alleles would be brown and blue. If the gene is hair color, the alleles would be brown or blonde or red, right? You see the difference between genes and alleles? Alleles are different versions of a gene, and you inherit one allele for a gene from one parent and a different allele of the gene from another parent, right? So sometimes they can say the same thing. They can be identical alleles. So for instance, brown hair and brown hair, you inherited the brown hair allele from both parents. But sometimes you might inherit different alleles from your two parents. You might inherit brown eyes from one parent and blue eyes from a different parent. You see the difference between the terms gene and allele? Hopefully this made sense. So what this should tell you is that each person has inherited two alleles for every gene, correct? Each person inherited two alleles for every gene. One allele from mom, one allele from dad. All right, hopefully everything is making sense so far and we fully understand what homologous chromosomes are. Remember, they are not identical. And we understand that there are 46 chromosomes in a typical human genome inside of each and every one of your cells. Now, you might be wondering, where are the sister chromatids? Okay, well, guess what? This cell with its 46 chromosomes, it doesn't have any sister chromatids. You want to know why? Because sister chromatids are exact copies of the chromosomes. And take a look here. There are only 46 chromosomes. Did you remember from chapter 8? Do you remember that before a cell divides, it needs to enter S phase of interphase? Hi, Wicket. Wicket's come to say hi, you guys. Um, and do you remember that during S phase of interphase, DNA is replicated? Do you remember that? Well, that's when we copy all the DNA. That's when the sister chromatids form. Only after S phase of interphase do we have sister chromatid pairs. So let me show you that now. Let's look at chromosome 1, right? Wicked scratching the monitor, you guys. So let's look at chromosome 1. And again, I'm just going to focus in on chromosome 1. Right now we have, homologous chromosomes from mom and dad. Now, take a look at chromosome 1. Let me zoom in. Let me take you to this picture here. Imagine if this was the unreplicated chromosome 1 with the maternal homologue on the left in pink and the paternal homologue on the right in blue. This is what it would look like in a skin cell. Let's say a skin cell that's not dividing. However, do you remember when the cell wants to divide, it passes the G1 phase, it passes G1 of interphase into S phase, the DNA gets copied. Okay, so basically what happens is we take the maternal chromosome on the left and we make a copy of it. So now we have two copies of mom's chromosome 1. You see that? Two copies of mom's chromosome 1. And we're going to copy dad's homologue as well. So we're going to get two copies of dad's chromosome 1 as well. And guess what? That's what the sister chromatids are. So these two pink ones are two exact copies of one another. They are two exact copies of, let's say, mom's chromosome 1. And then these two would be two exact copies of dad's chromosome 1. And so, these would also be sister chromatids on the right. And by the way, it is correct to say that these two blue ones, right? These two sister chromatids are homologous to the two pink ones, right? Because that means that They are the paternal on the right and the maternal on the left homologs for the same chromosome number. Okay, so all four of these are chromosome 1. There are four after S phase. After S phase of interphase, there are now four chromosome 1s, right? Four chromosome 1s, because you have two copies of mom's chromosome 1 and two copies of dad's chromosome 1. So you actually have four chromosome 1s. You'll also have four chromosome 2, four chromosome 3s, 92 total chromosomes inside of a cell, right? That's what happens by the end of S phase in interphase. So that leads us to clicker question number 2. Let's see if we can solve this together. The image below shows two chromosomes from one cell. Which statement is true? It looks like this is both of mom's chromosome 1, for instance. This is both of dad's chromosome 1. So we have four chromosome 1s, for instance. I'm just making that up. All right. A, both came from the person's mother. Did both of these come from the person's mother? No, it looks like these came from the maternal and these came from paternal sources, right? So A can't be the answer. How about B? The 1 to 2 pair is identical to the 3-4 pair. No, because 1 and 2 are identical. 3 and 4 are identical, but 1 and 2 are not going to be identical to 3 and 4 because they are different. They're from different parents. C, 1 is identical to 2. Isn't that the correct answer? Look at this. 1 is this sister chromatid is identical to the other sister chromatid. So the answer has to be C, and I hope you understand why. Again, these two, the 1 and 2 are sister chromatids, 3 and 4 are sister chromatids. However, 1 and 2 are homologous to 3 and 4. Now that we have a clear understanding of the difference between homologous chromosomes and sister chromatids, now we can really delve into the process of meiosis. Meiosis is how your sex organs, the testes or the ovaries, produce the gametes. Gametes are the sex cells. They include the sperm and the egg. Okay, so think of meiosis is how the sperm and the egg form. And by the way, did you know that the sperm and the egg, the gametes, are haploid? They are not diploid. Haploid means a cell that only has one set of chromosomes. So for instance, do you remember a set is 1 through 23, right? And diploid cells have two homologous sets of chromosomes. Well, a haploid cell only has one set. It does not have homologous sets of chromosomes. So essentially, a sperm would only contain 23 different chromosomes, and that's it, 23 total chromosomes, 1 through 23. And the egg contains another set, 1 through 23. And this is why during fertilization, which you can see here, the sperm cell is fertilizing this egg. And this, by the way, is an actual picture of a sperm fertilizing an egg with a scanning electron microscope. Isn't that cool? An actual picture of the process. The sperm, again, would contain 23 total chromosomes, 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, right? Do you recall? Those are called the paternal set of chromosomes. And the egg would contain the maternal set of 1 through 23 as well. And that's how you end up with the diploid zygote. Zygote is the fertilized egg. And again, meiosis is the process by which your body produces either sperm or eggs. So let's talk about that now. Again, for instance, if we look at these frogs here, the frogs undergo meiosis in order to produce the egg and the sperm. The egg and the sperm are haploid. And by the way, did you know that diploid cells are denoted as 2N for two homologous sets of chromosomes? So whenever you see 2N, you should know we're talking about diploid cells. And then the haploid sperm and the haploid egg are denoted as N. N represents one set of chromosomes. So the diploid parents produce haploid gametes. When fertilization occurs during mating, the haploid sperm fertilize the haploid eggs during fertilization to form the diploid zygote. Remember, zygote means the fertilized egg. The zygote is 2N. It has two homologous sets of chromosomes. And then this zygote divides by mitosis in order to form the tadpole, the juvenile frog. Again, this is a diploid juvenile, which undergoes more development and growth with mitosis in order to form the fully formed adult frogs. And by the way, did you know that the rest of the cells in your body are known as somatic cells? Somatic cells make up all the cells of your body except for the gamete cells, the sperm and the egg. Remember, the sperm and the egg are known as gamete cells, which are haploid. The rest of the cells in your body or in this frog's body are called the somatic cells, which are diploid. And by the way, did you know that meiosis only occurs in particular tissues in your body? Meiosis only occurs in the germ cells within the testes in males, or the germ cells in the ovaries within the females, right? So that's the only place meiosis is occurring. Diploid germ cells undergo meiosis to form the haploid gamete sperm and egg. So now that we know that meiosis results in the gamete cells, sperm and egg, and those sperm and egg cells are all genetically different. Isn't that interesting? One thing you should know is that meiosis undergoes 2 rounds of cell division. So I want to show you something. This cell on the left represents a germ cell. Do you guys remember the germ cells that are found in your reproductive organs? The testes or the ovaries possess these what are called germ cells. And you can think of germ cells as diploid or 2N cells, diploid cells. These germ cells are the ones that undergo meiosis to result in the sperm or the egg. Okay? And what did I just say? There's 2 rounds of cell division. So look at this. The germ cell divides once, okay, to form 2 cells. And then those 2 cells divide again to form 4 cells. So you should know that during meiosis, you form 4 genetically distinct, that means different, gamete cells after 2 rounds of cell division. Does that make sense? Make a note of that. That's different from mitosis. Do you remember mitosis resulted in 2 genetically identical cells after 1 round of division? Okay, meiosis undergoes 2 rounds of cell division, resulting in 4 genetically distinct gamete cells, which are haploid. Now, before we really delve deep into how each phase of meiosis works, let's take a quick break time with Gizmo and Wicket and see what these little guys are up to now. We'll be right back. All right, now let's look at each individual step of the meiosis process. The first thing that happens before meiosis can begin is a complete interphase cycle. Do you guys remember interphase is made up of 3 subphases, G1, S, and G2? Well, a germ cell with 46 chromosomes, let's say we're talking about a human cell, for instance, a germ cell is diploid and it has 46 chromosomes because it has 2 sets of homologous chromosomes. That cell in the reproductive organs will undergo interphase. And so during S phase of interphase, the DNA gets replicated. This means that you go from 46 total chromosomes to 92 total chromosomes because you've copied each and every chromosome, and now you have sister chromatids, and that's exactly what is depicted here in this cell that's in interphase. See, these two stringy red structures, this would be two copies of the same chromosome. So for instance, this would be two copies of mom's chromosome 1. Here's two copies of dad's chromosome 1. Here's two copies of dad's chromosome 2. And here's two copies of mom's chromosome 2. All right, so remember, now we have sister chromatids formed. This cell, if it was a human cell, after interphase would have 92 total chromosomes. It's ready to divide. Here you have the centrioles replicate during G2. Do you remember from the previous chapter that the centriole bundles replicate at G2 and they start to move to opposite ends of the cell? And that's exactly what happens before meiosis can begin. Again, interphase. The DNA replicates, the centrioles replicate, and we're ready for meiosis. And first, we're going to have Meiosis I, where there's one round of cell division, and then we're going to have Meiosis II, which is the second round of cell division, resulting in 4 total cells, gamete cells. Okay, so now we're going to talk about each individual step, right? So we're going to talk about Meiosis I. We're going to go into details about prophase I, metaphase I, anaphase I, and telophase I. And after that's finished, then we'll talk about the second round of cell division, Meiosis II, and we'll talk about how that works as well. You know, metaphase II, anaphase II, telophase II. We're going to go into those details as well. So let's break it down. All right, this image here depicts Meiosis I, the first round of cell division after interphase. Take a look here at the red box. The red box is showing you what's happening during prophase I. Now let's go into details here. All right, here is a cell in prophase I. Notice how you have the sister chromatid pairs, and those sister chromatids condense. Remember how DNA condenses when it's about to divide? When the cell is about to divide, I should say. So now you have condensed sister chromatids. And here's something interesting that happens during prophase I that I want you to know about. Not only do the sister chromatid pairs, right, both red ones, both blue ones, not only do sister chromatid pairs form, but the homologous pairs of sister chromatids all come together as a set of 4. You see this? This is known as a tetrad. A tetrad. Tetrads form. You know what a tetrad is? It's all 4 chromosome 1s, all 4 chromosome 2s, all 4 chromosome 3s. So all 4 of each chromosome will come together as a tetrad. Remember, tet means 4. And something special happens. These tetrads will undergo a process known as crossing over. Crossing over occurs, which means they will swap tails. You see these little tails here, the blue tail on this homolog and the red tail on this homolog? They're going to swap. So you're going to end up with swapped around tails. Take a look here. This is after crossing over has occurred on the tetrad. See, all 4 of these are chromosome 1. However, let me ask you this, and let me see if you can beat Wicket. After crossing over has occurred, are any one of these 4 chromatids identical anymore? That's right, Wicket. No, after crossing over, each one of these chromatids is now genetically distinct, genetically different. And I'm not sure why I didn't show crossing over on chromosome 2. Chromosome 2 would have crossed over as well. So you should see a little red tip here and a blue tip there. I'm not sure why they didn't draw that in here. But so again, let me show you something. This is what we need to know, right? During prophase I, tetrads form. That means all of chromosome 1 comes together, all of chromosome 2 comes together. So you're going to have, in a human, you would have 23 tetrads that form during prophase 1. And during prophase 1, what happens to these tails? What's that process called of swapping genetic information between homologous chromosomes? That's right, Wicket. It's called crossing over. And can you guess what is the purpose of crossing over? If you were to guess the purpose of crossing over, like why do cells cross over tails like this? What would you think? Can you beat Wicket? That's right, Wicket. To increase genetic variability. Do you remember that the purpose of sexual reproduction is to have genetically distinct offspring, right? Genetically different offspring. You want your offspring to be as genetically diverse as possible. That's what's going to give them the best chance of surviving, right? That ecological insult, right? So, again, what do I need you to know? I need you to know that during prophase 1, not only do sister chromatids pair, but then each sister chromatid pair also pairs up with the homologous sister chromatid pair as a tetrad. And then after tetrads form, crossing over occurs to increase genetic variability. So that's what I need you to know occurs during prophase 1. Next, we're going to move on to metaphase 1. During metaphase 1, homologous chromosomes line up in 2 rows. Another way of stating this is that the tetrads line up at the center of the cell. The tetrads line up. And I want you to notice something. Look at this. These tetrads line up in random fashion. So what do I mean? Look at this. Uh, if this is the tetrad here, I'm circling. If this is the tetrad for chromosome 1, it looks like both of the paternal homologs are lined up close to this pole, and both of the maternal homologs are closer to the bottom pole, right? However, the second tetrad, right, the tetrad for chromosome 2, is the opposite way. The maternal homologs are closer to the top end of the cell, and the paternal homologs are closer to the bottom end of the cell. And did you know that during metaphase 1, the tetrads could line up randomly, completely randomly? So, for instance, you could have had both of the paternal homologs pointing up for the tetrads, and both of the maternal homologs pointing down. Or you could have had what we see here, the blue one, the paternal homolog up for chromosome 1 and the paternal homolog down for chromosome 2. This is known as something you need to write down. This random assortment of tetrads during metaphase 1 is called independent assortment. And what does that refer to? It's the random orientation of tetrads during metaphase 1. There's like a 50-50 chance that they'll line up with the homologs on this side or that side, right? And that does what? Can you beat Wicket? What do you think is the purpose of independent assortment during metaphase 1? This random shuffling of tetrads during metaphase 1, how they align randomly. Can you beat Wicket? That's right. Wicket is always, it's to increase genetic diversity, right? To increase genetic variability. So this is something you need to understand about meiosis. During meiosis, there are 2 parts of meiosis that are responsible for causing all the genetic variation that you see in the gametes, right? The sperm and the egg. And those 2 important processes are crossing over, which occurs during prophase 1, and this random assortment, this independent assortment, which occurs during metaphase 1. So keep that in mind. That those are powerful things, and those are responsible for the massive amount of genetic variation that we see during meiosis. And then we're ready for Anaphase I. So let's talk about Anaphase I next. And look at this. During Anaphase I, homologous chromosomes separate. So The homologs are what separate from one another. Let me show you what that means real quick. So do you remember at the end of metaphase 1, the tetrads had lined up at the center of the cell randomly via independent assortment. So what's going to happen next is this. Look. The blue ones, you see, if I'm talking about the tetrad, the first tetrad here, the blue ones are going to separate from the red ones. Does that make sense? That's the easiest way of saying it. That means that the paternal homologs are separating from the maternal homologs. Both of dad's chromosome 1 separate from both of mom's chromosome 1. That's another way of saying it. So essentially, what they mean is that the homologs separate. The homologs separate. It's like this pair of sister chromatids separates from the homologous pair of sister chromatids. So you see what happens during Anaphase I? Look, the homologs separated. So if this was tetrad 1 on the left, you see what I'm circling? The paternal homologs are headed to this end of the cell, and the maternal homologs are headed to the other end of the cell. And yes, by the way, these aren't exactly sisters anymore, right? They're, remember these were called sister chromatids, but at this point they're called non-identical sister chromatids. You want to know why? It's because they're not perfectly identical anymore, and that's due to that crossing over that had happened during prophase 1. Okay, so you're going to do what during anaphase? Can you beat Wicket? That's right, Wicket. During anaphase 1, homologs separate. Homologs separate. You can also think of it as pairs of sister chromatids, non-identical sister chromatids, separate from homologous pairs of non-identical sister chromatids. And let me ask you this and see if you can beat Wicket. If this is true, if this is how they split, are these two cells going to be identical at the end of meiosis I? What do you think? Take a close look and tell me, are the two cells that are about to develop, one up here and one down here, are these going to be genetically identical cells? Can you beat Wicket? No, of course not. That's right, Wicket. No, these are not going to be identical cells, right? Because think about it. This cell is going to get both of dad's chromosome 1 and both of mom's chromosome 2. Whereas this cell is going to get both of mom's chromosome 1 and both of dad's chromosome 2. So these are actually completely different cells, right? Genetically speaking, you're already ending up with two genetically distinct means not identical cells at the end of meiosis I. And that's exactly what happens during telophase. You see, a daughter nucleus forms here, a daughter nucleus forms here. And notice how you end up with two cells, and those two cells are not genetically identical. All right, so we are now at the end of meiosis I, the first round of cell division of meiosis. But remember, we still have not formed the gamete cells, the sperm or the eggs. So we're ready to leave meiosis I and enter meiosis II. So here we go. So each one of these two cells that just formed is going to divide again. So that's, see, this cell's going to divide again, and this cell's going to divide again. All right, so let's take a look at how this works. During prophase 2, you should know that, I'm going to zoom in a little bit more for you. Okay, during prophase 2, the non-identical sister chromatids form. And you could call these sister chromatids, but technically these are non-identical sister chromatids which form, because remember, crossing over has left them not identical. Next, during metaphase 2, what lines up at the center of the cell? Yep, it's those non-identical sister chromatids which line up at the center of the cell. And then during Anaphase II, what separates from what? The non-identical sisters separate from one another. Non-identical sister chromatids separate from one another. And then in telophase 2, daughter nuclei form and cytokinesis occurs. And that results in, finally, we have our four genetically non-identical haploid daughter cells called the gametes, the sperm, and the egg. And remember, haploid is just denoted as a regular N, not 2N. So again, this second round of division is called Meiosis II, and it results in the gametes for genetically non-identical gamete cells, right? The haploid cells. Now, before I move on to the next slide, you should realize that now these haploid cells, they're haploid, so they have one complete set of chromosomes. What does that mean? That means each one of these should have 23 total chromosomes. That means only one chromosome 1, one chromosome 2, one chromosome 3, and so on, right? 23 total chromosomes in each of these gamete cells. And remember, each gamete cell, each sperm, each egg is completely genetically unique. There are no two sperm, no two eggs that should have the same genetics. So each sperm and each egg is incredibly unique. All right, this leads us to clicker question number 3. A zygote, remember a zygote is a fertilized egg. A zygote of a ferret contains 40 chromosomes. How many chromosomes are in a ferret's sperm cell? Well, remember, a zygote is diploid, which means it has 2 complete sets of chromosomes. So if a zygote has 40 chromosomes, that means the sperm and the egg should only have 20. Does that make sense? So the answer is 20. And you should already know this slide, right? Take a look at this slide. Meiosis generates genetic variability. The chromosomes in the gametes produced during meiosis are different because of what? Remember, what were the 2 sources of genetic variability during meiosis? Remember, during prophase 1, we had crossing over, and during metaphase 1, we had independent assortment. Those are powerful. Those are responsible for all the genetic variation in the gamete cells. In fact, here's a closer look at crossing over. During crossing over, 2 homologous chromosomes pair up, right? Remember in the tetrad, they form the tetrad, and exchange pieces, scrambling the genetic material. So here's the tetrad. Let's say this is the tetrad for chromosome 1. So all 4 of these are chromosome 1s. Remember, during prophase 1, they come together to form a tetrad. And then in that tetrad, the little tail swap over, right? So the little blue tail swaps to this sister, and the little red tail swaps to the other sister. This is known as crossing over, which forms. And now you have what? You have 4 genetically different chromatids, don't you? Because do you remember these 2 blue ones? These 2 blue ones were sister chromatids. They were identical. See, they were identical. But after crossing over, they are no longer identical. In fact, they are genetically non-identical. And so are these 2 sisters down here, right? You see, these are 2 sister chromatids for mom's chromosome 1. And notice that after crossing over, these are also not identical. So again, you see how crossing over in the tetrads during prophase 1 results in genetic variability. And that, look, when these sisters end up in a gamete, let's say sperm cells, notice that each sperm cell, right? Remember 4 sperm cells form? Each sperm cell is going to get a different genetic makeup. You see, this sperm cell's got the solid blue chromosome. This this sperm cell gets the the paternal chromosome that's been crossed over. This one gets the red chromosome that has a crossover, and this one gets a solid red chromosome. So the main point is you end up with gametes that are all genetically different, all genetically unique. And that's this strong. If crossing over didn't happen, by the way, you guys, if crossing over didn't happen, well, then these two cells might be the same, right? These two cells would be identical, and these two sperm cells would be identical. But because of crossing over, they're not going to be identical. Isn't that neat? And then also, do you remember I told you that independent assortment makes for genetic variation as well? And that's because these tetrads line up randomly inside of the, inside of the cell. So look, if we have three different tetrads, right? Look, tetrad 1, 2, and 3, the tetrads could line up like this during metaphase 1, correct? Or they could have lined up like this. Look, this is alternative 2. This is just as likely to occur as this one, right? But notice how that's what independent assortment is. It's the random assortment, the random orientation of the tetrads during metaphase 1. So look at alternative 1. If the tetrads had lined up with all of the paternal homologs pointing up, after meiosis, you would see that one cell would get all three of the paternal homologs, whereas the other cell would get all three of the maternal homologs. Whereas if during metaphase 1, if the result had been alternative 2, well, then after meiosis, this gamete cell would have received the paternal homologs for chromosome 1 and 2, but the maternal homolog for chromosome 3. And conversely, this gamete cell would have received the maternal homologs for chromosomes 1 and 2, and the paternal homolog for 3. Notice that this is a different genetic makeup down here than for up here. You see, so independent assortment makes for more genetic variation as well, doesn't it? And here's some more alternatives, right? This could happen as well. Those three chromosomes could have lined up like this, which would have given you this result. Here's another alternative. They could have lined up like this and given you this result. I want you to understand all four of these are equally likely to happen, giving you distinct outcomes that are all genetically different. You see the power of independent assortment? It's going to give you all kinds of rearrangements of these chromosomes in your offspring. Now, the result of crossing over and independent assortment are massive amounts of genetic variability in your offspring. But invariably, students often ask me, but Dr. D, aren't twins identical genetically? How does that work? If it's so rare, if there's so much shuffling that happens with the sperm and the egg, with the genes and the genetics, how come twins have the same exact DNA makeup? Well, let me explain that real quick in this next slide. Take a look here. Monozygotic twins are genetically identical. So what is monozygote? Mono means one zygote twins. So what does that mean? Let me show you. In this case, with identical twins, this is how it works. A single sperm, look at this, a single sperm fertilizes a single egg to give you the fertilized egg called the diploid zygote. And that zygote divides by mitosis to give rise to the embryo. And at some point, part of that embryo pops off and it actually forms two embryos. Isn't that weird? So look, a single zygote somehow ends up kind of pinching off into two embryos. And so wouldn't those two embryos have the same genetics because they came from a single sperm and a single egg, which is really interesting, right? Because if you think about it, if you know any twins or if you are an identical twin, they were roommates, right? They were the ultimate roommates. They both shared this one room or cell, at one point in their history. Isn't that amazing? So both people were one cell at one point. And that single zygote with its genetic makeup popped apart, into two different embryos, and that resulted in identical twins that have the exact same genetic makeup. Isn't that interesting? And now you understand why it's called monozygotic twins, because they literally came from one zygote. However, you may have heard of fraternal twins, right? These are called dizygotic twins, fraternal twins. Like, so if you've ever heard, someone having twins, but one's a boy and one's a girl, and they're not genetically identical, how did that work? Well, in this case, there were two different sperm which fertilized two different eggs, and that led to two different zygotes. That's what dizygotic means. Di means two zygote twins. So obviously, each one of these sperm would have its own genetic makeup. Each one of these eggs would have its own genetic makeup. So yes, you're going to end up with two embryos and two offspring, but those offspring are going to be completely different genetically. Isn't that interesting? So fraternal twins come from two sperm and two eggs, whereas identical twins come from one sperm and one egg. And that's the big difference there. All right, and this leads us to our next clicker question. Clicker question number 4. Many sets of triplets, this is when you have three children at once, consist of a pair and a spare. That is, two identical babies and one non-identical baby. Producing this arrangement requires how many sperm and how many eggs? Well, think about it. For the two identical babies, you would only need one sperm and one egg. But for the non-identical baby, you would need another sperm and another egg. So the answer should be two sperm and two egg. That's the correct answer. And so what have we learned? Haven't we learned that mitosis and meiosis are really quite different? In mitosis, the cells underwent interphase, copying all the chromosomes. Then during prophase, do you recall during prophase, the sister chromatid pairs formed. During metaphase, sister chromatid pairs lined up. And during anaphase and telophase, the sister chromatid pairs separated and daughter nuclei formed, giving you what? Two genetically identical daughter cells. And that was it. Do you remember? That was mitosis. Cell division resulting in two genetically identical cells. But right now we learned about meiosis, which starts with interphase. The DNA gets copied. Next is Meiosis I. During Meiosis I, we have prophase I, where tetrads form and crossing over occurred to increase genetic variability. Metaphase I, where the tetrads line up randomly, right? By what process? Can you beat Wicket? What was the random process by which tetrads line up at the center of the cell during metaphase I of meiosis? Can you beat Wicket? That's right, Wicket, as always. Independent assortment. Independent assortment, which increases genetic variability. And then we have anaphase 1 and 2, anaphase 1 and telophase 1, and that results in two genetically unique cells. And then we have metaphase 2, where the non-identical sister chromatids line up, and then anaphase 2, telophase 2, where those non-identical sister chromatids separate, and this forms four genetically non-identical haploid cells called the gametes, the sperm and the egg. Now that we understand meiosis, let's talk about some errors that can occur sometimes during meiosis. If chromosomes fail to separate properly, abnormal gametes form. This failure to separate is called nondisjunction. Here you can see nondisjunction in Meiosis I. Let me show you this. Let's take a close look at this. Here is a diploid cell. What they're showing you here is a cell, a diploid cell, a diploid germ cell. And let's say this is the tetrad. You see, this is the tetrad. Let's say tetrad for the 21st chromosome. The 21st chromosome, okay? So imagine at the end of meiosis I, two of those chromosome 21s should have ended up in this cell, and two chromosome 21s should have ended up in this cell down here. But what happened? Notice how all four chromosome 21s ended up in this cell, and zero chromosome 21s ended up in this cell. Well, do you know what that means? That means that by the end of meiosis II, the gametes, instead of each gamete having one chromosome 21, notice how these two gametes have two chromosome 21s, whereas these two gametes have zero chromosome 21s. Do you see the problem there? That's nondisjunction. These chromosomes did not separate correctly. The homologs did not separate correctly during meiosis I. And what happens during fertilization? Take a close look here. During fertilization, the sperm will bring with it a single chromosome 21. So if this sperm fertilizes this egg, you'll end up with two maternal chromosome 21s and one paternal chromosome 21. Same thing with this egg. However, these eggs down here, these eggs would only have one paternal chromosome 21. Do you see the problem here? The problem is you're going to end up with something called aneuploidy. Aneuploidy are gametes with abnormal numbers of chromosomes, right? So, when you have, for instance, these zygotes, this zygote's going to have three chromosome 21s. This zygote's going to have three chromosome 21s. These zygotes are going to have one chromosome 21. You see how that's an abnormal number of chromosome 21s? A normal cell has one uh, paternal homolog for chromosome 21 and one maternal homolog for chromosome 21, right? So when you have non-disjunction and you have improper separating of the chromosomes during either meiosis I or meiosis II, this can result in aneuploidy with abnormal numbers of chromosomes in the gametes and abnormal numbers of chromosomes in the zygotes. Okay, and here is an example of non-disjunction during meiosis II, anaphase II. So in this case, look, it's a little bit different, right? In this case, the diploid germ cell, it divided correctly. The tetrads separated correctly during meiosis I, see? However, it was during meiosis II that the sisters didn't separate correctly. You see that? So you end up with the same problem. Like I said, whether non-disjunction occurs during meiosis I or meiosis II, you're going to end up with aneuploidy, this abnormal number of chromosomes in the gametes, and that's going to result in abnormal numbers of chromosomes in the zygote, in the offspring. So for instance, this offspring, this zygote has too many chromosome 21s, whereas this one has too few chromosome 21s, whereas these two zygotes would have the correct number of chromosome 21s in this scenario. And actually, that's exactly what causes Down syndrome. Down syndrome is caused by individuals who have three copies of chromosome 21 rather than the traditional two copies of chromosome 21. So if we look at their karyotype, again, remember a traditional karyotype is two of every chromosome. See, two of every chromosome. However, people with Down syndrome have three chromosome 21s. It's called trisomy 21, tri meaning three. And that leads to Down syndrome, right? And now you understand the cause of Down syndrome, either non-disjunction in meiosis I or non-disjunction in meiosis II. Either way, this can result in abnormal numbers of chromosomes, leading to various genetic disorders. In fact, it's interesting, non-disjunction of sex chromosomes causes various sex disorders. So for instance, you can have three X chromosomes. You remember the sex chromosomes X and Y? Traditionally, a male has an X and a Y chromosome, and a female has two X chromosomes, but it is possible to be trisomy for the X chromosome, so you can have three X chromosomes instead of two. This is known as triple X syndrome. It happens in 1 in 1,500 females. It leads to a tall stature, menstrual irregularities, increased risk of giving birth to triplo X daughters or even XXY sons. Isn't that interesting? And there are other ones as well. We're not going to talk about each and every one, but for instance, you can have two X chromosomes and a Y chromosome. This is known as Klinefelter disease or XXY syndrome. It happens in 1 in 750 males. It's variable symptoms, but often includes sexual underdevelopment, long limbs, large hands and feet, development of breast tissue. So we can see that there are so many different types of aneuploidy, so many different types of extra chromosomes that can occur, too few chromosomes that can occur that result in various genetic abnormalities. And this leads us to clicker question number 5. In Turner syndrome, a female is born with only one X chromosome. That's it, just one X chromosome. This disorder could arise as a result of nondisjunction in what? The sperm, the egg, or either the sperm or the egg. It could be in either, really, the sperm or the egg, though nondisjunction is more common in the egg. And again, just to recap, meiosis produces the human gametes, the sperm and the eggs. Germ cells located in the testes give rise to the sperm cells, while germ cells located in the ovaries give rise to the egg cells. And notice just how much smaller a sperm cell is than an egg cell. An egg cell is several times bigger than a sperm cell. All right, and that leads to the end of chapter 9. I hope everything made sense. As always, I do have a link here. I'm going to link it right above here. You can see the card above to my longer explanation of meiosis for my 4 science majors course. If you want to watch that for even more details, you can if you like. I hope it all made sense. Let me know in the comment box below if you have any questions, and I will catch you guys next time.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. A Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D.